Welcome to the RNA-Seq data analysis, downstream data analysis, and biological interpretation workshop. This exciting 3-day journey is designed to equip you with the essential skills needed to analyze RNA-Seq data and uncover meaningful insights. Whether you are a beginner or you're looking to refine your skill, this workshop has something for everyone. Let's see, for the day one, we have data preparation, normalization, and dimensionality reduction as a goal. So day one sets the stage with a focus on these topics so that we have a solid foundation for the downstream RNA-Seq analysis. We'll start by diving into the RNA-Seq data formats, and we'll explore how to extract the data sets from the repositories like GEO, NIH, TCGA, then we have Firebrowse, then NCBI, SRA, then Expression Atlas, ENA, GPR, and the Human Protein Atlas. The next, we are going to tackle the dimensionality reduction, which is a very essential step for making sense of high dimensionality RNA-seq data. From principal component analysis, multidimensional scaling, we are going to talk about the advanced techniques like t-SNE, and you will get the hands-on experience on visualizing, clustering, and identifying sample patterns. So by the end of the day one, you'll have the confidence to prepare and normalize RNA-seq data and reduce its complexity for insightful analysis. For the day two, we are going to delve deeper into differential expression analysis and pathway enrichment. For DNA expression analysis, we are going to talk about the DSEC2 package on the Collab, and we are going to introduce some statistically concept of p-value, FDR, and log-fold change. Next, we are going to talk about the visualization of the differential expression of the results. We are going to create the volcano plots, the MA plots, and the heat maps by using various R libraries on the Collab itself. And then we are going to move towards the pathway enrichment analysis, where we are going to talk about various tools like KEGG, Reactome, EnrichR, and Links. And here we are going to prepare some plots also for the visualization of the results for the biological context here. Now, by the end of the day two, you will be well versed in identifying differentially expressed genes and understanding their role in biological pathways. Now, for the day three, we have planned for protein-protein interaction network and hub gene analysis. For this, we are going to talk about the online database STRING. And then we are going to get a hands-on experience on the Cytoscape. And further, we'll take the Cytoscape for the validation of and identification of the hub genes. And we are going to take the hands-on experience for some other plugins like CytoHoba and MCode for the further analysis. So we'll see by the wrap-up of this workshop, we'll talk about all the integration, and you'll be equipped to analyze the complex network and identify the key genes and draw meaningful biological conclusion. So this workshop is your gateway to mastering the downstream RNA-seq data analysis and biological interpretation. So join us and unlock the potential of your research.